Hello, everyone, and welcome to Semel Grand Rounds. Today, we are very pleased to be featuring Dr. Jennifer Forsythe, giving us a talk about finding the path from genes to the clinical presentation in schizophrenia. If you have questions for Dr. Forsythe, feel free to ask at any point using the Q&A button, either during the talk or at the Q&A at the end. And a final reminder, just to make sure to do your CME credit survey that will open up either right after the talk, or you'll get an email in your inbox tomorrow around noon. And with that, I will turn it over to Dr. Nectarline to introduce the speaker.
Thank you. I am very pleased to introduce Jennifer Forsythe, who's currently an assistant professor in our Department of Psychiatry, and this summer will be transitioning to the University of Washington Department of Psychology, where she's starting a tenure-track assistant professor position. We'll miss her, but we're glad that we're able to hear her grand round before she leaves. Dr. Forsythe completed her PhD at UCLA in the Department of Psychology, Clinical Psychology program, did her internship at the University of Pittsburgh, then returned to UCLA where she was a T32 postdoctoral fellow for 2 years, and then was awarded a NIMH for Development Award, as well as a NARSAD, Young Investigator Award. She was selected in 2017 by the Association for Psychological Science for a Rising Star Award. And their citation said it recognizes outstanding psychological scientists whose innovative work has already advanced the field and signals great potential for their continued contribution. That's very fitting for Dr. Forsythe. It has been my pleasure to watch her in recent years develop her program of research in which she's analyzing genomic data in conjunction with clinical, neurocognitive, and structural imaging data to identify biological pathways associated with psychosis and psychosis-related phenotypes. And she'll be speaking to us today about that topic. So without further ado, Dr. Forsythe.
Thank you so much for that kind introduction, Keith. Yeah, it really is a pleasure to be invited to speak at Grand Rounds today. So let me go ahead and share my screen. Okay. Yeah, so as Keith mentioned, I did my training here at UCLA, the vast majority of it, and it really has been just a fabulous environment for me in so many ways to both understand the richness of the clinical phenotype of schizophrenia, and in recent years to really start to take advantage of a lot of the huge technological advances that we've had in genetics and functional genomics. And I'm really so grateful for all of the training that I've had here over the years. Okay, and so today my talk is going to be mostly geared around some of my more recent work, and the title is Finding the Path from Genes to Complex Syndrome in Schizophrenia. And just, you know, I think given this crowd, people probably understand why there's an urgent need still for us to do more in terms of understanding schizophrenia, but it is a debilitating psychiatric disorder. And from a clinical perspective, it's been largely defined by the presence of psychotic symptoms like hallucinations or delusions that typically emerge in late adolescence or early adulthood. And despite decades of research by a lot of really brilliant scientists, there's still a lot that we don't understand about the core biological processes that underlie the disorder. And in many ways, our current treatment strategies are still not effective enough to prevent the decline in functioning and disability that affects so many patients. So for example, rates of unemployment are extremely high. Up to 90% of patients are unemployed or make less than minimum wage. About 65% of patients are severely impaired in terms of social functioning. Many of them are really pretty socially isolated. And overall, patients have a reduced lifespan by about 10 to 20 years, which is partially due to high rates of suicide, which are highest in the first few years after symptom onset. And so I think this tells us that really there's an urgent need for us to do more and keep pushing forward to try to find new ways to improve patient outcomes and to understand the etiology of this disorder. And when I think about this, I think there's a number of key facts that are really important to keep in mind. So first off, it's a broad syndrome. And so while we've typically focused on the presence of psychotic symptoms to diagnose the disorder, it's also associated with various sensory, motor, and cognitive signs and symptoms, many of which emerge prior to psychotic symptom onset. And the cognitive impairment in schizophrenia is now generally considered a core component of the disorder. It's present prior to psychotic symptom onset. It may be the most stable and debilitating feature of the illness, and it's also one of the best predictors of long-term outcome. And that series of findings related to the really key role of cognitive impairment in schizophrenia was established in large part through seminal work by folks here at UCLA like Keith Nectarline and Michael Green. And from imaging, EEG, and postmortem brain studies, we also know that there are disruptions within and across many brain regions and neural circuits in schizophrenia. And we know from family studies that it's a highly heritable disorder. The heritability rate has pretty consistently been estimated around 80%, which has told us for quite a while that genes play a major role. And finally, there's also a lot of clinical and biological heterogeneity between patients. So while we see deficits and abnormalities across a range of these different domains, the differences are seen to at varying degrees from one patient to the next, and patients have different clinical trajectories and presentations. And for me, these have always really seemed like a sort of set of puzzle pieces that we need to make sense of and try to put together. If we want to really understand the core mechanism or mechanisms that lead to this syndrome and how we can best improve patient outcomes. And so a little about my own path and the history of the field. When I started in this field, maybe about 13 or 14 years ago as an undergrad, at that time, we didn't have genes or genetic variants that were reliably associated with schizophrenia. And we didn't even really know much about the genetic architecture of the disorder, like how many genes might be involved or what kinds of variants were involved. And what we did have were some major etiological models. So for example, at the time, the Nmda receptor dysfunction model was a fairly dominant model that suggested that the major molecular dysfunction underlying this complex syndrome was hypofunction at this receptor. And a lot of that came out of findings that, for example, if you blocked the Nmda receptor with drugs like ketamine or PCP, that could produce positive, negative, and cognitive symptoms in healthy individuals that were really strikingly similar to what we saw in patients with schizophrenia. So really recapitulating many aspects of this schizophrenia syndrome. And among patients with schizophrenia, these same drugs would exacerbate symptoms. There were also some postmortem brain findings and early genetic findings that also implicated the Nmda receptor. And so as an undergrad, I started off working on these Nmda receptor antagonist models of schizophrenia. And I was really fascinated by the fact that disrupting signaling at this key glutamate receptor could reproduce so many aspects of the syndrome, including, for example, the dopamine dysfunction that we know is a key target of antipsychotic medication. And over the years, I'll say, I've been less convinced by this model as a complete explanatory model. Part of that is because, some different studies that aim to use the Nmda receptor or other glutamate receptor agonists to try to improve cognitive and positive symptoms in schizophrenia, they had some inconsistent or to me, which felt like also somewhat disappointing effects. And I also saw that during some work that I did during my PhD. And also there was this nagging thought that I had that, we've known for a while that a lot of the signs and symptoms associated with schizophrenia seem to emerge prior to psychotic illness onset. And this Nmda receptor antagonist model really didn't address that. And also in recent years, there's been these huge technological changes, and with the advent of large-scale genetic studies, especially in the last 5 to 10 years, we've started to have robust genetic associations with schizophrenia. And in these studies, some of the genes that have been implicated are outside of the Nmda receptor complex. So that suggests that Nmda receptor hypofunction model is probably too simplistic. However, for me, this overarching idea of trying to identify some convergent molecular dysfunction signal that could lead to broader neural system dysfunction and ultimately the complex syndrome is something that has really stuck with me. And so now, as I'm thinking more about genetics with these recent major gains, I think that this is a really helpful anchor point, right? Where now we can start at the beginning of this path to try to understand how we get from genes to complex syndrome and schizophrenia. So for today, I'm going to tell you about some recent work that I've been doing that leverages these gains in genetics to try to understand this path. And first, I'll talk about some work trying to understand the relationship between risk for schizophrenia and brain development. I'll then talk about some work to try to understand some of the clinical heterogeneity that I alluded to that we see among patients, including thinking about schizophrenia in the context of one specific genetic syndrome, 22q11.2 deletion syndrome, as well as trying to understand the heterogeneity among patients more broadly. Okay, so first, thinking about genetic risk for schizophrenia and brain development. So with this rise of really large-scale genomic studies in the last 5 to 10 years, such as those through the Psychiatric Genetics Consortium that often involve tens of thousands of patients and controls, what we now know is that genetic risk for schizophrenia is highly polygenic, so it likely involves hundreds to thousands of genes. Common and rare variants confer risk, including de novo mutations and copy number variants, as well as some other classes of variants. Common variants account for the largest proportion of genetic liability for schizophrenia when we think about the whole population of patients, so about 30 to 50% of liability, but each individual common variant has a really small effect. And on the other hand, deleterious rare variants account for a smaller proportion of risk when considered across all patients, but when they're present in a given individual, they can really increase risk substantially. And that's what this figure here is illustrating, where the frequency that a variant occurs in the population is marked along the x-axis, going from really low frequency on the left to more high frequency or common variants on the right. And the increase in risk associated with a variant is shown on the y-axis. And so the blue dots are common variants that have been associated with schizophrenia, which have a teeny tiny effect. And the green dots, for example, in the upper left of the plot, are showing, for example, rare copy number variants, which, as you can see, for some of them, when they do exist in a given patient, they can really increase risk substantially. So taking a step back to make sure we're on the same page about what I mean when I'm talking about these different kinds of genetic variants. So common variants are just variants that are found in more than 5% of the population. Rare variants are ones that are defined usually as being found in less than 0.5 to 1% of the population. De novo mutations are newly arising mutations that are found in a child that did not exist in either parent. And copy number variants are a special kind of genetic variant that occurs when a large chunk of DNA is deleted or duplicated. There are also some other more complex classes of copy number variants, but I'll just talk about these more straightforward deletions or duplications today, which have been pretty robustly now associated with schizophrenia. Okay, so again, coming back to this model where if what we want to understand is that path from genes to molecular dysfunction to broader neural system dysfunction to this syndrome, these genetic findings allow us to ask on a systematic genome-wide basis, do risk variants for schizophrenia converge onto specific neurobiological processes? And if so, to what extent does this occur across common and rare variants? And more specifically, like I mentioned before, with so many of these subtle signs and symptoms occurring in schizophrenia, having their onset long before psychotic symptoms actually emerge, this has for quite some time suggested that schizophrenia is most likely a neurodevelopmental disorder. So if we're trying to understand how genetic risk for schizophrenia might impact the brain, I think the critical question is really, how does genetic risk impact brain development more specifically? Okay, so then I think it's important to ask what is happening during normal brain development. And we can think about brain development as divided roughly into two major periods. So the first is fetal brain development, which is shown here on the left, where the primary thing that's happening is the initial assembly or formation of the brain. And this is an intricate process in which billions of cells have to proliferate, differentiate from embryonic stem cells into neurons and other brain cell types. They then have to migrate to their proper position in the brain and form initial synaptic connections with one another. And then during postnatal development, the big task is really refining and reorganizing all of these neurons and initial connections into a mature brain that works precisely and is maximally effective in our environments. So around birth, we know that there's an overproliferation of neurons and synapses, and it's kind of like all these cells and connections are auditioning and training for their roles, and the ones that are working well, they stick around, they fine-tune their signaling and become stronger, while the others get pruned away, which is what we often talk about as synaptic pruning. So what makes this big orchestrated process of brain development actually unfold? For example, what makes this embryonic stem cell turn into a neuron or a different cell type? All of this is happening through changes in gene expression. And one thing to keep in mind is, of course, that all the cells in our body contain the exact same DNA, which can be divided into blocks of sequence that code for about 20,000 different proteins in the human body. And because each gene gets transcribed one at a time into messenger RNA, which is what you can see here, which then gets translated into the proteins that form the building blocks of all of our cells and tissues. It's this process of the selective expression of different combinations of genes that tells one pluripotent stem cell to turn into one specific brain type or another. And overall, this is what drives the process of brain development. And in line with this, changes in gene expression are especially prominent during early brain development, when all these cells that will make up the brain are first proliferating and differentiating into their eventual cell type. And so this fact about gene expression and these changes in gene expression driving being really dynamic during brain development in order to drive development creates a really useful property of gene expression data, in that genes that operate together to drive specific processes or are expressed to give a cell a stable identity have similar patterns of expression, which allows them to be clustered or organized by their co-expression into the biological systems in which they operate. And this is work that was pioneered by the Horvath lab here at UCLA. And so another way to think about this kind of clustering is that if we were to think about, for example, all first-year graduate students at UCLA and take pictures of who was in what building on a given day over the years, and if each of these circles is a student and the lines or edges represents how often they're showing up in the same place with each of the other students, we could probably do a pretty good job of figuring out the students who were psychology grad students, for example, versus public health students, versus medical students, or dental students, and business school students. And when it comes to brain development, again, where gene expression is changing really dynamically, this kind of clustering technique can be a really powerful strategy for organizing genes into the developmental, cellular, and molecular systems in which they operate. And so following some really seminal work by the Geschwin lab, also here at UCLA, that first used early pre and perinatal brain samples from the publicly available BrainSpan data set, which is a data set that essentially measured genome-wide expression of genes across brain regions in the human brain during development. The Geschwin lab first used that data set to understand convergent risk for autism. In this context, here we leverage BrainSpan samples spanning late embryonic development through 30 years of age to capture neurodevelopmental processes across the full span of brain development. And what this output was 18 modules across over 17,000 genes that captured broad aspects of neurodevelopment. And each of these modules ranged in size from about 50 to 3,000 genes. We validated these modules for shared biological function using other data sets and found that yes, they were enriched for gene ontology-based sets of genes that are known to be involved in shared biological or molecular functions. They were enriched for co-regulation by transcription factors, so they're expected to be co-expressed together. And they were also enriched for genes that are known to operate together in protein interaction networks. So altogether, this suggested that yes, these modules represent real aspects of biology. We also found that the modules were enriched for known markers of specific primary brain cell types. So here, this is showing each of the modules on the x-axis and major brain cell types on the y-axis. You can see that there were several modules that were enriched for genes that are known to be expressed specifically in neurons. That's that first row. Other modules were enriched for markers of other brain cell types, such as an oligodendrocyte specific module, M10. And some modules weren't enriched for markers of any cell type. And we could then ask which of these modules are associated with genetic risk for schizophrenia. So to do that, I compiled summary statistics, variant and gene lists from the largest genome-wide studies of schizophrenia in the literature at the time and asked which modules are enriched for these risk variants. And just looking at these genetic risk data sets a little more closely, they were studies that involved thousands to tens of thousands of patients and controls. And they're listed here from data sets identifying common to more rare and copy number variants associated with schizophrenia. So what did we find? Again, with the modules on the x-axis and each of these data sets going from common to rare and CNV data sets on the y-axis, if there's an asterisk, that means that the module was significantly associated with the data set after correcting for the number of modules. And if there's a plus sign, that means that the module was nominally associated with the data set before correcting for modules. And both are marked here to give you a sense of potential convergent enrichment across common and rare data sets. So there were 3 modules that came up as significantly associated with schizophrenia. This one, M15, which was enriched for common and copy number variants associated with schizophrenia. M13, which was also enriched for common variants. And M7, which was enriched for common variants with some enrichment for rare variants as well. And so looking at what each of these modules are doing, starting first with M15, because it was really highly enriched for common schizophrenia variants as well as CNVs associated with schizophrenia. This was a neuronal module, like I mentioned earlier. And the plot on the left is showing the expression trajectory of M15 across development with expression level on the y-axis and age on the x-axis starting at 6 weeks post-conception. There is, by the way, I'm so sorry, there is a vacuum cleaning operation happening in the hallway of my building right now. So I'm so sorry if that is noisy, hopefully not too disruptive. Anyways, back to M15, this module, this neuronal module. Right, where the x-axis is showing age across development of the samples, and the y-axis is the amount of expression. You can see that this is a module that increases in its expression across development. And the plot on the right is showing the top terms that were associated with genes in this module, the p-value on the x-axis. And so the genes in this module were broadly involved in synaptic signaling, refinement of neurotransmission, and learning and memory. And now looking at the M13 module that was enriched for common variants associated with schizophrenia. This was also a neuronal marker enriched module that showed increasing expression during prenatal development before plateauing around birth. And this is a module that was involved in basic aspects of synaptic transmission, such as ion channel activity and regulation of the membrane potential, indicating that this is a module that was involved in regulating overall neuronal excitability. And finally, looking at module 7, which showed some enrichment across common and rare variants and was not enriched for any specific cell types. This is a module that showed its highest expression during early prenatal development and subsequently came down in its expression, but its genes were generally expressed across development. And this is a module that was involved in regulating the expression of genes from DNA to RNA, indicating that this module was involved broadly in regulating gene expression across development. So altogether, this suggests that at the patient population level, schizophrenia risk variants converge on gene networks that are involved in establishing neuronal excitability and in executing the workhorse neuronal function of synaptic transmission that we often think of, and that is also inherently linked to the refinement of neurotransmission and neural circuits during postnatal development. And one question that I had, given my own research history was, where are the Nmda receptor complex genes relative to these modules? And sure enough, it did turn out that the neuronal M13 and M15 modules that were associated with risk for schizophrenia were enriched for genes that are part of the Nmda receptor complex. However, if we consider the whole synapse, which is what this figure is showing, individual genes that were associated with schizophrenia, yes, they impact the Nmda receptor, but they also impact other ion channels and receptors in the synapse, proteins that hold the pre and postsynaptic cell together, and scaffold proteins that anchor receptors and other proteins in the postsynaptic density, as well as other intracellular signaling proteins involved in translating receptor activation into the changes that then result in refinement of synapses and synaptic transmission. So altogether, this suggests that risk variants for schizophrenia impact neuronal excitability and synaptic signaling pathways that are intrinsically linked to synaptic refinement during postnatal development, during a time when environmental input is especially critical for shaping the synaptic pruning that drives neural circuit maturation. And that this dysfunction in synaptic refinement across circuits could lead to the emergence of the broad schizophrenia syndrome across development. Now, one other set of findings that were also coming out around this time in the psychiatric genetics field is that variants that confer risk for schizophrenia might partially overlap those involved in earlier onset neurodevelopmental disorders, such as autism and intellectual disability. So this raised some additional questions. Specifically, do risk variants for earlier onset developmental disorders like autism and intellectual disability converge onto specific neurobiological processes? And if so, are the associated developmental processes similar or distinct from the ones that were associated with schizophrenia? So we then compiled, similarly, the largest genome-wide association studies of common and rare variants for autism. And we also looked at de novo mutations for intellectual disability. Since this class of mutations has been strongly associated with intellectual disability. And we asked which modules were associated with risk for these earlier onset disorders. And so again, here with the modules on the x-axis and the variant data sets on the y-axis for schizophrenia, autism, and intellectual disability, he found that the same modules that came up for schizophrenia showed some enrichment for autism and intellectual disability risk variants. But there were also 4 additional modules that were more distinctly associated with autism and intellectual disability. And of these additional modules, I'm going to focus on just a couple here that came up really robustly as associated with autism or intellectual disability. So starting with M1 here, this is a module that showed its highest expression during early to mid-fetal development and came right down in its expression afterwards. And in fact, this module was highly enriched for genes that are expressed specifically during fetal development. And the genes in this module are involved in regulating transcription and chromatin organization. So really, this module appears to be involved in orchestrating the gene expression changes that are critical for early brain formation. And this other module that was robustly associated with autism and intellectual disability risk variants was M4, which is a module that was highly enriched for neuronal markers and peaked in its expression during mid to late prenatal development. This is a module that was involved in neuronal differentiation and morphogenesis, or the outgrowth of axons and dendrites that form synapses. So the genes in this module appear to be critically involved in the early development of neurons and neural circuits. And indeed, there is likely a link between these 2 modules, as the most connected hub genes in the M1 module actually are transcription factors that are known to promote neuronal differentiation from embryonic stem cells. And I'll say that these findings of these developmental enrichment pattern of genes that are associated with autism and intellectual disability, as well as these biological pathways that were associated, are ones that have now been pretty reliably, robustly found across many different studies trying to understand convergent biological processes involved in autism spectrum disorder. Okay. So overall, what we were seeing was that while schizophrenia, autism, and intellectual disability risk variants each converge on postnatal aspects of synaptic transmission and refinement of synaptic transmission, risk for autism and intellectual disability seem to additionally converge on networks involved in neuronal differentiation and morphogenesis during fetal development. And so coming back to this model, where it looks like one primary path from genes to complex syndrome in schizophrenia may be altered neuronal excitability and synaptic signaling, which ultimately disrupts the activity-dependent refinement of synapses and circuits during postnatal development, while genes that lead to earlier onset neurodevelopmental disorders such as autism and intellectual disability are additionally impacting early neuronal differentiation and morphogenesis, leading to a disruption in the initial formation of neural circuits during prenatal development, as well as disrupted circuit refinement. Okay, so that sort of summarizes some of the findings if we're thinking about risk variants that have been associated with schizophrenia and earlier onset disorders at the population levels. But again, as I was mentioning earlier, there's a lot of heterogeneity between patients. And I think this is really important to understand, and to understand how that heterogeneity might intersect with brain development, because we want to know what biological processes to intervene on for a given patient, when is the optimal time window for intervention, and more specifically, what that looks like for each individual patient, so that we can optimize intervention for individual patients who might have different genetic risk profiles. So essentially trying to move towards a personalized medicine approach. So as we've been talking about, the genetic architecture of schizophrenia is complex, and it involves common, rare, de novo, and copy number variants. However, the specific genetic variants that confer risk for one patient can vary dramatically from the next. For example, some patients might have a risk loading profile that largely comes from common variants, whereas another patient might have a highly penetrant copy number variant that is driving the majority of their risk. And we also know that there's a lot of heterogeneity in terms of clinical presentation between patients. So for example, some patients have comorbid learning disabilities, autism spectrum disorder, or intellectual disability in childhood. There's also variability in the age of psychotic symptom onset and in the overall trajectory of functioning between patients. And in the literature, there have been hints that there could be some meaningful structure to this heterogeneity. So for example, some studies have found that if you cluster patients based on their cognitive developmental trajectory, 3 groups sometimes come out that differ in terms of their age of onset, severity of symptoms, and cognitive profile. So for example, sometimes these studies will see a stable poor group who might have poor cognitive functioning beginning in childhood that stays poor through adolescence and adulthood, a stable good group who might have average or above average functioning in childhood and remain in that average range throughout adolescence and adulthood, and also a deteriorating group who start in more of the average functioning range in childhood but show a decline over adolescence. And interestingly, one recent structural MRI study found when they looked at these kinds of cognitive trajectory groups, that the stable good group had milder reductions in gray matter volume, whereas the deteriorating group had more dramatic reductions in gray matter volume that were most prominent in frontotemporal regions. So recapitulating sort of what we think about as a typical pattern of gray matter deficits in schizophrenia. And in this study, unfortunately, they didn't have a large enough sample size to look at the stable poor group. And in the genetics literature, there was also a recent paper that came out that found that schizophrenia patients who have comorbid intellectual disability, so that's the group on the left of this plot, were more likely to have rare loss of function variants in genes that were associated with more severe neurodevelopmental disorders. That's what's shown by that burgundy dot on the left of the plot, compared to patients depicted on the right who don't have comorbid intellectual disability. And thinking some more about this heterogeneity, I think it can be useful to think about the overall pattern of cortical changes that we see in schizophrenia in a scenario where there's an unknown genetic etiology, or what we sometimes call idiopathic schizophrenia. So this plot on the left is showing the effect size differences from a sample of 173 schizophrenia patients and 128 demographically matched controls. And to orient you to the color scale, the regions in dark yellow are near 0, while cooler colors that are more green or blue are regions where patients show reductions in cortical thickness or surface area. Warmer colors would be regions where patients show an increase compared to controls, but as you can see, that's largely not seen in idiopathic schizophrenia. And cortical thickness and surface area are what make up cortical gray matter volume, which is shown on the right, where cortical thickness is the thickness of the 6 cortical layers, which is thought to be influenced by synaptic density and intracortical myelination, whereas cortical surface area is the total area covered by cortex for a given region, and that's thought to be influenced by cell cycle parameters, including the balance between cell proliferation and apoptosis during early corticogenesis. And so in idiopathic schizophrenia, like I mentioned, the largest reductions in cortical thickness occur in frontotemporal regions, while reductions in surface area are milder overall. And what do the cortical changes look like in the context of a highly penetrant genetic variant? So for example, if we come back to this figure showing the increase in schizophrenia risk associated with different variants, you can see that one variant that increases risk substantially is copy number variants at 22q, at the 22q11.2 locus, specifically deletions at this locus. And so for most individuals who have this deletion, it spans a region of chromosome 22 that encompasses about 45 protein coding genes. And I was really fortunate during my postdoc to work with Carrie Bearden here at UCLA, who has done a lot of really seminal work to try to understand this copy number variant and how it confers risk for psychosis and other disorders. And so this is a variant that is one of the most highly penetrant individual risk variants for schizophrenia. It's found in about 0.3 to 1% of schizophrenia patients. However, patients with the deletion also frequently have an IQ that's in the borderline intellectual disability range, and they also have elevated rates of autism and ADHD, as well as mood disorders. And it also seems like there's some preliminary work that suggests that this deletion may be more common in patients with childhood onset versus adult onset schizophrenia. And if we look at the pattern of cortical changes in deletion patients with psychosis, it actually looks really quite different. So patients with 22q deletion have a thickening of cortex in the majority of regions, although they still show thinning in some regions associated with schizophrenia more broadly, like temporal cortex regions. And there's also a really prominent reduction in surface area across regions. So their brains are overall smaller than controls. And notably, these reductions in surface area are actually nearly twice the magnitude in terms of effect size relative to the cortical thickening pattern. And if we look at which brainspan modules the deleted 22q genes are in, yes, there are genes in a number of modules that were associated with schizophrenia, such as M15 and M7, but there's also 22q genes, for example, in the M1 module that was associated more strongly with autism and intellectual disability. This suggests that there might be important aspects of the background genetic profile of these patients that could contribute to some of the differences in the phenotype that we see relative to idiopathic schizophrenia. And so in one recent study that we did to try to identify which genes within the locus might drive these major brain phenotypes in 22q, this is difficult to do experimentally in humans, but one approach that's become possible in recent years is to use comprehensive maps of gene expression from the human brain, such as the Allen Human Brain Atlas, which is another publicly available transcriptomic data set that measured the expression of over 20,000 genes in postmortem brain tissue in healthy individuals at a really high spatial resolution. And then to look for correlations between the patterning of variation in gene expression across regions relative to variation in some sort of brain imaging phenotype of interest. And these kinds of studies have found, for example, that brain regions that are closer in physical proximity or functionally correlated activity share more similar transcriptomic expression patterns. And in clinical populations, this kind of approach has been used to try to hone in on molecular mechanisms that might underlie brain phenotypes. So for example, in Alzheimer's, a recent study found that variation in amyloid pathology and APP expression were found to be correlated, and also that variation in neurodegeneration and MAPT expression were also correlated. So when we applied this as part of the 22q Enigma consortium, so we integrated gene expression data from the Allen Human Brain Atlas and looked at the severity of cortical changes in terms of surface area and thickness deviation in patients with 22q deletions compared to controls, and asked whether variation in severity of deviation was associated with the expression of any 22q genes. What we found was that the expression of DGCR8 was significantly associated with the pattern of surface area reductions. And notably, DGCR8 is a gene within the 22q locus that is known to be intolerant to loss of function. And it's located in the M1 module that was associated with autism and intellectual disability. It's a gene that's known to play a major role in regulating diverse gene expression patterns during early brain development because it's a key component of the protein complex that creates a class of non-coding RNAs called microRNAs that target the transcripts of other genes to repress their translation into proteins. And these microRNAs are indeed known to be really important for regulating gene expression in the brain during fetal development, including for regulating cell proliferation and differentiation. And so our finding that DGCR8 might be a potential major contributor to brain changes in 22q deletion syndrome is consistent with some growing literature from animal and brain-based organoid models of 22q, which have also highlighted disrupted microRNA biogenesis due to DGCR8 haploinsufficiency as a likely contributor to brain phenotypes in 22q 11.2 deletions. And so taken together, as we think about this specific CNV as one example, so we know like I was saying, that this deletion is something that greatly increases risk for schizophrenia, but it is also associated with autism and a borderline IQ profile. And there are really pervasive reductions in cortical surface area in patients with this deletion, which we don't really see in idiopathic schizophrenia. And the 22q11.2 gene DGCR8, which is a gene that was in that M1 associated in that M1 module that was associated with autism and intellectual disability, seems like it could be a major contributor to brain phenotypes in the deletion. And that's not to say that other genes within the deletion might not also be playing a role. They likely are, but I think it's really interesting that there's a growing base of findings suggesting that this may be a really critical gene for a lot of the phenotype that we see in the deletion. Okay, so then if we're coming back to this model again, where a common pathway to schizophrenia involves genes that disrupt neuronal excitability and synaptic signaling that impacts the refinement of synapses and circuits during postnatal life. And that might be most strongly shaped by common variants of small effect, and a common path to earlier onset developmental disorders, such as autism and intellectual disability, includes risk variants that disrupt neuron differentiation and proliferation during fetal development, in addition to alterations in neuronal excitability and signaling, a greater loading of which is likely due to larger effect de novo mutations or CNVs. We might think about this clinical picture with psychosis onset in late adolescence or early adulthood with mild cognitive deficits as maybe a typical patient with psychosis, whereas some patients might have additional risk loading from genes that are more typically associated with autism and intellectual disability, and that might involve greater disruptions to earlier aspects of neuron and brain formation. And that could lead to a phenotype that more commonly involves a borderline IQ or intellectual disability in an earlier age of illness onset. And it seems like 22q patients with psychosis could be an example of that. While other patients might have a lower overall loading of risk variants for schizophrenia, possibly a higher contribution from environmental factors such as substance use or trauma. They might present with average or above average functioning and be a patient that we might consider to have good prognostic features. So these are some of the hypotheses that I'm really interested in currently, and I'm trying to explore from a more systematic perspective. And that's what my K award is focused on. So in the last few minutes, I'll just wrap up talking about this project and some initial findings that we're seeing. So going forward, a key question that I have is whether differences in the genetic architecture or loading of genetic risk within specific neurobiological pathways, whether these differences might associate with different clinical phenotypes among patients that then also suggest different treatment targets. So for this K award, I've been so fortunate to have my mentors and collaborators here at UCLA, folks that have done really seminal work on schizophrenia over the years, who were willing to share their data that they've collected over years, where they've really comprehensively characterized the phenotypes of different psychosis populations, and also where they collected DNA so that we could start to ask these questions. So the samples for my K were coming from the labs of Dr. Carrie Bearden, Dr. Keith Nectarline, Dr. Robert Asarno, who was also my PhD advisor, and Dr. Michael Green. And these clinics were focused on either adolescent onset psychosis, recent onset adult psychosis, childhood onset schizophrenia, and chronic schizophrenia patients. And this is work that's very much ongoing, but in the initial batch of DNA samples that we've started to process, we have about 400 patients with schizophrenia. We also have some relatives and some controls in there. I'll mention from a clinical perspective, I find it interesting, for example, that cannabis use disorders are higher in the patients with schizophrenia compared to the relatives and controls. And in this sample, we do have a pretty wide range of age of onset in terms of age of onset of psychotic symptoms. And we do see, the typical pattern where schizophrenia subjects in this sample have a lower overall IQ compared to their relatives and controls. And for this project, so far what we've done in terms of looking at genetic variants is we've called rare protein altering variants. So essentially, we used exome sequencing and genome-wide genotyping to call single nucleotide variants and small insertions or deletions, as well as calling copy number variants using the genotyping data, so that basically we can then look at the overall risk loading among these patients of deleterious variants that essentially are either protein truncating, so they're expected to really reduce the expression of a given gene, either because they're protein truncating, or similarly, if it's a copy number variant, there's a deletion of a chunk of DNA, so that would also reduce the expression of that gene. And we're focusing so far mostly on rare variants because these are more likely to actually have a deleterious effect. And just some very preliminary findings so far. What we've seen is that the overall burden of rare deleterious protein altering variants in this schizophrenia cohort among the patients that have a higher burden of these deleterious variants, either across all genes or across genes in the M6 module that was associated with autism, or autism-specific genes, having a higher burden of deleterious variants in these genes was associated with a higher likelihood of having an IQ in the borderline range. There were also some nominal associations with other modules. That's if there's a little cross, then it's a nominal association not corrected for the number of modules. If there's an asterisk, that means it is corrected for the number of modules, or the comparisons that we did here. So you can see that there are also nominal associations with other modules that were associated with autism, such as M1 and M13, which was also associated with schizophrenia. And now, one thing about this sample is that there were 9 subjects that were included in this sample that we knew at the time when we included them that they had a 22q deletion as well as a psychosis diagnosis. So one thing we wanted to ask, since that's a little bit more of a homogeneous sample, is do we still see this signal if we exclude those subjects? And we do still see some hints of signal. So removing that subset of patients, we did still see some increased burden in autism-associated genes with the likelihood of having an IQ in the borderline range. And if we look more specifically at rare deleterious protein-altering variants in genes that are known already to be intolerant to loss of function, again, with all subjects in the sample, increased burden among M1 genes was associated with borderline intellectual functioning. And there were also nominal associations with the total number of genes as well as genes in M13. And again, if we remove the known 22q psychosis patients, that does weaken some of the signal, although there is still, interestingly, a burden of deleterious protein altering variants in known loss of function intolerant genes in M1 genes, which is that module again that was associated with autism and schizophrenia risk, as well as schizophrenia associated genes. So far we are seeing some very preliminary support for this idea that patients with poorer functioning might have a greater burden of rare protein altering variants in genes that are associated more so with autism and intellectual disability. And there's a lot more to do on that project in particular. We're looking at other phenotypes. We are also refining our variant calls, and this summer we're going to be adding another large set of patients. And looking forward, I think some critical next steps that I'm really interested in is thinking about longitudinal studies starting in childhood, where we can prospectively assess the relationship between different kinds of genetic risk profiles and the emergence of different clinical, cognitive, and neurobehavioral signs and symptoms across development. I think we can also use this kind of a framework and apply it to other disorders to start to understand how genetic risk profiles sort of shape the development of these clinical trajectories and different outcomes over time. And, you know, I think ultimately what we're most interested in the long run is the hope that we can develop biologically informed interventions. And in many ways, I think we already have an arsenal of tools that can be useful for many patients. But if we think about the trajectory of onset and intensity of psychotic symptoms that emerge in, you know, late adolescence or early adulthood for most patients, where major aspects of brain development are occurring, you know, understanding whether someone has a genetic risk profile that you know really is impacted by variants that are you know that impact genes that are critical for later versus earlier aspects of brain development, I think can give us really helpful clues about when our major windows of opportunity for intervention are for different patients and what specific interventions we should try to employ. Okay, so with that, I realize I've run a little over time, so I just want to thank, you know, the many people who have mentored me throughout my career, including my K mentors, most recently for some of this new data, Carrie Bearden, Roel Ophoff, Keith Nectarline, and Dan Geschwind. Giovanni Coppola was also a postdoctoral co-mentor, and Robert Asarno, who was my PhD advisor. I also want to thank all the folks in the Bearden lab, the Ophoff lab, and my many collaborators at UCLA and elsewhere. The UCLA Aftercare Research Program and CAPS program, who've done so much of the data collection, and my funding agencies. Okay.
Thank you very much, Jen. That was a fascinating talk and really covered a lot of territory. One question we might have time to address is Gil Hoffman asks, you showed enrichment of the schizophrenia risk genes loading onto postnatal processes. How can you reconcile that with the evidence from recent studies suggesting expression of schizophrenia risk genes in the placenta, as well as numerous prenatal environmental risk factors for schizophrenia?
Yeah, that's a great question. So I think, I mean, so first, let me say that regarding the second part of the question about prenatal environmental risk factors that have been associated with schizophrenia, you know, here in this talk, I've really focused on, you know, how can we use genetic risk variants to try to understand the etiology of the disorder. But I really, you know, haven't been talking about these environmental factors, many of which, you know, there are many different environmental risk factors that we know impact risk for schizophrenia, and likely many of which interact with some of the genetic risk variants. So I think that if we were to incorporate prenatal environmental risk factors, I think we might see that, for example, folks who do have, you know, maybe obstetric complications or hypoxia around birth, those might be folks that might have a lower, you know, overall genetic risk loading for schizophrenia. That's speculative, but I think that's a really interesting question. And I also think some of the genes, or like, for example, there's that M7 module that's involved, that was associated with schizophrenia and autism risk, that's associated with regulating gene expression across development. I think it's interesting thinking about the, about like the placenta findings, for example. Are some of the genes that are implicated or that might be relevant for placenta findings, are they genes that actually are involved otherwise in gene regulatory functions? And so, I think we don't know. And I think, that would take looking at placenta tissue separately to try to understand. Yeah, so I think that this is just one way of trying to understand some common biological processes, but certainly this is not, encompassing absolutely everything. There's a lot of other data to try to incorporate to understand both, the role, the absolute role of these environmental factors and also how they interact with genetic risk.
So thank you very much, Jen. I think we need to stop. We're at the top of the hour. That was a great talk and was educational for those who don't know very much about the genotypic ways of looking at schizophrenia, and really helped us track conceptually through the whole process of trying to translate from genotypes to phenotypes. So thank you very much. We look forward to following your work at the University of Washington.
Thanks so much. Thank you everyone for having me and for coming.